All right, so our final chapter that we'll be covering in Bio 2010 is chapter 14, which is all about the eukaryotic cell cycle and the process of mitosis and meiosis. I will begin today's discussion covering section 14.1 from your book, and we'll begin with a brief overview of key features of the chromosomes. So this mini video is just on chromosomal features. So just to give you context, what we're talking about is when we have a cell and that single cell is going to become two cells. And we're focusing just on eukaryotic cells. We're not going to talk about binary fission, which takes place in prokaryotes. But again, we are wanting to focus on what's happening and what's required when one cell becomes two cells. And in that discussion, it's important for us to differentiate between two main classes of cells. We either have the somatic cells, which are all the cells that comprise the cells in your body, with the exception of your reproductive cells. The reproductive cells are your gametes or your sex cells, and that would either be egg and sperm. So when we're talking about the somatic cells, what we're going to focus on is the process of mitosis, whereas when we're talking about the gametes, the egg and the sperm, we will go through the process of meiosis. And those two processes have a lot of overlap, but they also have some unique features to them. So we will walk our way through that carefully. When we're talking about egg and sperm, it's really important to recognize that each of them is going to have 23 linear chromosomes. And when those two come together, we are going to form what we call a diploid cell, or the zygote, which is also known as the fertilized egg. And so when the fertilized egg is formed, we're going to have one entire set of chromosomes coming from dad, and one entire set of chromosomes coming from mom. So depicted here is the sperm and the egg, and then we're going to have the fertilized egg. Again, just the nuclear DNA is going to be contributed to the egg from the mom, and so again, you're going to be inheriting mitochondrial DNA from your mother, but you'll have within the nucleus an entire set of chromosomes coming from dad and an entire set of chromosomes coming from mom. And each of those chromosomes are going to make up, each pair of chromosomes is going to make up what we call our homologous pair. So just to orient you, this is an older picture of my own 3 kids. This was taken quite a while ago, but I wanted to utilize it to illustrate a point. So the previous chapter, we talked all about gene expression, and really when you're expressing a gene, what you're doing is you are ultimately going to be making a protein. And so the definition of a gene is just a unit of nucleic acid that specifies the inherited trait of an organism. And so how is that actually expressed or presented? We call the character whatever is the observable heritable feature. And the trait is kind of the version of that particular character that you can see. So for instance, my daughter Anna takes after her dad and has very blonde hair, whereas I have dark hair. So we would say that Anna's gene that codes for hair color, and hair color would be the character, specifies the trait, which is blonde. And so that's kind of how we would utilize this terminology. So we want to make sure that we're differentiating between genotype and phenotype. Genotype, again, is the makeup of all the genes for a particular individual, and we've talked repeatedly about the fact that any cell in your body is going to have the same genotype as another cell in your body. However, what proteins are being expressed there is going to vary, and so those proteins being expressed is going to contribute to the overall function of a particular cell, and because based upon protein expression, we will that will kind of determine the phenotype, which is the physical manifestation of those genes. That's going to be what we're able to see and observe. All right, so when we are talking about genes, again, these are distinct portions within a chromosome, and it's a very specific sequence of nucleotides that confers a particular trait. So again, here we would have a chromosome, say, from dad and another chromosome from mom, and we can look on the chromosomes and identify the exact location for a particular gene. All the genes have now been mapped onto human chromosomes, our 23 different chromosomes, and we have, again, one from mom and one from dad. And then we can actually look at that sequence and see how are they similar and how are they different. So an allele is a version of that gene, but there can be multiple alleles for a particular gene, and this is why we have such variation in terms of things like eye color and height and even personality traits, extroversion, depression, anxiety, ADHD. These have all been mapped onto our chromosomes. There's some important nomenclature to fully understand when we're having a discussion about chromosomes. So the first thing is that by convention, it was determined that the largest chromosomes would have the lowest number. So again, chromosome 1 is going to be our largest chromosome, whereas chromosome 22 is our smallest chromosome. Notably, chromosome 1 is going to have a lot more genes on that chromosome relative to chromosome 22. Our sex chromosomes are, by convention, denoted as the last two chromosomes. So if you have two X's, that would be female. If you have an X and a Y, that would be a male. We also have, relative to the centromere, which is shown here as this yellow circle, we also have what is known as the short arm and the long arm. The short arm is known as the petite arm, and so we call that the P arm. And then the Q arm, so right after P comes Q, that is by definition our long arm. And so you can actually map genes and determine its location. So for instance, we know that on gene 9q34, so we could go to 9q34, this banding pattern right here would depict the genes that confer your particular blood type. And so you can actually go on, our government has websites where you can go and look at the physical location of basically every gene that has been sequenced and mapped onto chromosomes. So again, our eukaryotic cells, with the exception of the egg and sperm, are all diploid, meaning they have one copy from mom and one copy from dad. And for a particular gene, you can look at the form of that gene, the form that came from dad and the form that came from mom. And so again, here we're saying this particular chromosome, the form of the gene at this location came from mom, and relative to the form of the gene that came from dad. And so if those two forms of the gene are identical, we say that it's homozygous, whereas if the form of the gene is different, we say it's heterozygous. And if you continue on and take genetics, we will explore the idea of genes being dominant versus recessive, and how that plays into your overall phenotype. So we actually can do a karyotype for individuals, and also you do excessive staining, and so we have this particular banding pattern, and you can figure out what is going on in terms of the genes for a particular individual. So go ahead, pause the video, take a look at this, and see what might be concerning for this particular karyotype. All right, so first of all, I hope that you were able to observe that we have two X chromosomes, so this would be a female. However, we see 3 copies of chromosome 21. So this would be, for instance, how one might identify an individual who has Down syndrome, which is called trisomy 21, 3 copies of chromosome 21. Most chromosomes, if you have a trisomy, 3 copies of that entire chromosome, it's actually will not be sustained in pregnancy, And so, for instance, if you had 3 copies of chromosome 1, because there are so many genes that are being expressed, the body would not tolerate that. So we only see trisomies going to term with the shorter chromosomes. We sometimes see it with chromosome 18 and 21 especially, and that is better tolerated. So we'll go ahead and stop our discussion on chromosome structures here, and I'll pick up in the next video talking about the actual cell cycle.